Scientists just used a 3D printer in space, but they did not print a plastic toy. They printed tiny structures made with living kidney and liver cells. Why travel hundreds of miles above Earth to do something that sounds like it belongs in a hospital? The strange answer is gravity. A small bioprinter aboard the International Space Station made structures containing human kidney, liver, and cartilage cells. It also made 28 small nerve repair implants. The samples returned to Earth, where researchers can study how well they formed. This is a real first. But let's make one thing clear immediately. Scientists did not print a complete kidney or liver ready for transplant. They printed small pieces of living tissue. That difference matters. A normal 3D printer builds an object one thin layer at a time, often using melted plastic. A bioprinter follows a similar idea, but its ink can contain living cells mixed inside a soft supporting gel. Imagine building a brick wall. The cells are like the bricks. The gel is like the mortar that holds the bricks in place. The printer follows a digital map and places the mixture in a planned shape. The goal is not merely to make something shaped like body tissue. The cells must stay alive, organize themselves, receive food and oxygen, and eventually do a useful job. That is much harder than printing plastic. On Earth, gravity is always pulling materials downward. If you leave a glass of muddy water alone, the heavier pieces slowly settle toward the bottom. Living cells inside printing material can behave somewhat like that. They may sink, spread, or become uneven before the structure is stable. Earth-based printers can use thicker gels or temporary supports, but those supports may make printing more complicated and can affect delicate cells. On the space station, everything is still under Earth's gravity. The station and everything inside it are falling around Earth together. That creates microgravity, the floating effect astronauts experience. With much less settling, cells may remain more evenly spread while the printer builds the tissue. Think of trying to build a tower from soft jelly. On Earth, it sags. In microgravity, the builder gets a different set of rules. Kidneys clean waste and extra water from blood. The liver processes nutrients, handles chemicals, and performs hundreds of jobs. Both are complicated organs filled with different cell types and tiny pathways. Because they are so complex, they are useful tests for bioprinting. Researchers can ask, did the cells stay alive? Were they spread evenly? Did the layers keep the intended shape? Did the cells begin acting like the tissue they came from? The returned samples must now be examined. Printing is only the beginning. Testing tells researchers whether the result is truly useful. The nearest uses may be smaller than a full organ and still important. Scientists could make tissue models for testing medicines. A tiny liver-like sample might show whether a drug damages liver cells. Tissue patches might someday help repair part of an injured organ. The same printer may also produce medical implants, such as guides that help damaged nerves grow in the right direction. If microgravity consistently improves these products, future commercial space stations could become unusual factories for certain medical materials. Space manufacturing is expensive. Every machine, cell sample, and supply must survive a rocket launch. Finished products must return safely. Researchers also need to prove that space-made tissue is better enough to justify the cost. Then come the usual medical hurdles: repeatable manufacturing, safety tests, clinical trials when appropriate, and review by regulators. So this experiment does not mean hospitals will order space kidneys next year. Full transplantable organs need blood vessels, nerves, many kinds of cells, and a way to connect safely with a patient. Scientists are still far from solving all of that. Important inventions often begin as small demonstrations. The first airplanes carried very few people. Early computers filled rooms and did less than a phone. Their importance was not that they were already perfect. They proved a new path could work. This experiment shows that one compact machine can make several kinds of biological material during one trip in orbit. Researchers now have real samples to measure, compare, and improve. The honest headline is not, "Scientists printed replacement organs in space." It is. Scientists took another step toward learning whether space can help us manufacture better living tissue. That is less magical, but much more interesting, because it is true. The next question is what the returned samples reveal. Are the cells healthier? Are they more evenly arranged? And is the improvement worth launching a factory into orbit? What should we explain next? A robot that can fly and swim, or tiny robot boats that join together like ants? Tell us below. This is Breakthrough Explained. Big discoveries, simple explanations.